So in this video we're going to read about the anatomical structure of the roots. First of all, we're going for the monocot root. So the anatomical structure of the monocot root, it is slightly different, not slightly, a bit more different than the stems. So when we cut the TS of the monocot root, it is also circular, and first I'll draw the circular structure and then I'll go for the description of each and every part. So when we cut the root transversely, it means we take a TS section of the root. We find that it is circular and it has many hairs, and obviously root will have the hairs for the absorption of water and minerals. But as of stem, there are no double layer. In stem we had the epidermis and then we had the hypodermis, but here only single layer is there. Yeah, but inner one is slightly similar, two layers are there. And now the arrangement of the vascular bundles in root. Now remember, in root there is no conjoint vascular bundles. Vascular bundles are conjoint only in the stem. In root, radial type is present. It means that the xylem and phloem, they represent alternately at different radii. It means if this is the xylem, then there is phloem, then xylem, then phloem, and xylem, then phloem. So it is somehow like this. Okay, so this layer, it is known as epiblema. In root it is known as epiblema. No epidermis, epiblema. The term to be remembered, epiblema, is in root. These are the root hairs, and again these are the same endodermis. This is the Peri Cycle, this is the cortex, this is the pith, these are the vascular bundles. Now one more thing to remember, that in the stem we saw that these layers between them, between the vascular bundles, they were known as medullary rays, but here they are not known as medullary rays, they are known as conjunctive tissues. Conjunctive tissues. So epiblema and conjunctive tissues are two different terms used in the roots. So this is basically the TS. Now we're gonna describe from the outermost region to the innermost region. So first of all, we'll describe about the epiblema. So while describing about the epiblema, there is not much to write as of the stem. In stem we just wrote that the epiblema, the epidermis, it is the outermost layer composed of the parenchymatous cells, and it is covered with cuticle. And also in the dicot we told you that the stem has multicellular hairs. But now while writing about the epiblema, see, it is also the outermost covering. It is made up of compactly arranged parenchymatous cells. But now the epiblema, it consists of hairs which are unicellular. The hairs of the dicot stem, they are the multicellular hairs, and the hairs of the monocot root, they are unicellular hairs. This is the basic point of distinction. It has unicellular hair and stem has multicellular hair. So it has unicellular hair. Okay, it has unicellular hair, and yeah, cuticle is not present in epiblema. Cuticle is only present in epidermis, no cuticle. These are the point to be remembered in the epiblema. And then we'll go on for the cortex, because there is no hypodermis. So basically the cortex is very similar to the cortex of the stem. So it is also a mass of it is also the mass of cells which are lying beneath the epiblema. The round parenchymatous cells which are thin walled, having less intercellular spaces. So there are also the parenchymatous cells which are rounded or oval in structure, lying below the epidermis, but here lying below the epiblema. So cortex is almost similar to the cortex of the stem. Now after cortex we are going for the endodermis, the inner cell wall. So even here the endodermis is the innermost layer of cortex. In stem we saw that the endodermis, it was the innermost layer of the cortex, which was formed of somewhat barrel like cells which were thickened because the starch grain was present there. If you forgot, you can go to my last video, the part 2. So in this endodermis, it is also the last layer of the cortex, and even it is formed of barrel shaped cells. Now what does barrel shaped cells means? It means that if this is the cortex, then the epidermis is somewhat like this, somewhat rectangular. So these are known as barrel shaped cells. Actually barrel shape is somewhat like this. So they are formed of barrel shaped cells, and yeah, it is also thickened. The cell wall is thickened, though it is also made up of parenchymatous cells, because it is the last layer of cortex. So obviously Cortex is a parenchyma, so endodermis will also be of parenchyma, but it is thickened. Now that in stem the thickening was due to the deposition of starch grains, but here the thickening is not due to the deposition of starch grains, it is due to the deposition of the waterproof strips which are known as lignin and suberin. Now the thing I'm going to tell you, it is one of the most important and almost fixed question for the very short marks questions, for the 1 marks questions. Now I'll tell you about the Kasparian strips and passage cells. In the endodermis of the root, these cells, these endodermis, they have some strips. Okay, these strips are due to the deposition of some lignin and suberin. Remember the name, lignin and suberin. These are deposited in the endodermis, and so these strips are present. They are waterproof strips, so the water cannot enter inside this endodermis. But some cells of the endodermis, like this cell, it lacks the suberin and lignin. It means they do not have the Kasparian strips. Now these cells which do not have the Kasparian strips, they are known as passage cells. And why they are known as passage cells? Because the water can enter through this. These are waterproof, so water cannot enter. But this cell, it does not have the Kasparian strips, so water can enter. So they are known as passage cells. And about the Kasparian strips and the passage cells, it is regularly asked in the 1 marks question. So basically endodermis, we can write about it for more information, like it is the last layer of cortex in form of barrel shaped cells. It is thickened due to deposition of lignin and suberin, so they contain Kasparian strips. But some cells they allow the water to pass, known as passage cells. So this will be enough for endodermis. Okay, and then we go for the innermost layer, which is known as Peri Cycle. So Peri Cycle, now it is also very similar to the Peri Cycle of the stem. It is also the innermost layer lying below the endodermis and above the vascular bundles, as we wrote in the stem. But yeah, now in stem the Peri Cycle was slightly confusing, because it was Sclerenchymatous above the vascular bundle and Parenchymatous above the medullary ray. You can see my last video. But here there is no such controversies. The Peri Cycle is simply, it is formed of Parenchymatous cells. It is simply formed of parenchymatous cells. Remember, Peri Cycle is parenchymatous in the root, and the Peri Cycle, it is Sclerenchymatous along with the parenchymatous in the stem. So only the thing confusing here is somewhat endodermis. That's also not much confusing still. Now after the Peri Cycle, we can see the vascular bundles are present there. So we'll write about the vascular bundles. So though we need to write about the vascular bundles in detail, but it is one of the most easiest to write, because we just need to write which type of vascular bundle is present, and then xylem and phloem. So it's very easy to write, because we can see the figure and distinguish what type of vascular bundle is present. So when we cut the TS, we see that the vascular bundles are arranged alternately at different radii. It is here, and this is at this radii, this is at this radii. It means different radii, and alternately xylem, phloem. So this type of arrangement, it is known as radial arrangement. I have taught you this in the part 1 of this video. So vascular bundles, they are radial type, and since it is radial type, the xylem and phloem, they are arranged radially at different radii. So this term is not necessary to write. Xylem and phloem are simply situated at different radii, and alternately you can write just the description of the radial type, just to make it slightly longer. So after writing this, just write about the xylem and phloem. And yeah, one more thing, it is characterized with 6 more than number. This xylem and phloem, they are more than 6 in number. It means it can be 3 xylem and 5, sorry, not 3 xylem, it can be 3 xylem and 3 phloem, because the xylem and phloem are equal here. Okay, remember, xylem and phloem are always equal in radial type. So it can be 4 xylem and 4 phloem, but it is always more than 6. The total number of vascular bundles, it is more than 6. So this is not an important info to write, still I told you. So after writing radial type, write about the xylem. So xylem, now here comes the point of distinction. In both of the stems, we saw the xylem was endarch. It means the protoxylem was lying towards the inner side and the metaxylem towards the outer side. But here the xylem is exarch in root. Okay, the xylem is exarch. Now what that means, the protoxylem, it lies towards the outer side and the metaxylem towards the inner side. If this is the pith here, so metaxylem will lie towards the pith and this protoxylem, it will lie towards the pericycle, towards the periphery. So it is exarch type, and then you can write that it is made up of tracheids, vessels, xylem parenchyma, xylem fibers, and then phloem. Even here you can write that the phloem, it is present in between the xylem, and it is made up of phloem parenchyma, phloem fibers. It is made up of sieve tubes, companion cells, just the formation. So with this, vascular bundles are also over. Then in STEM we used to write after the vascular bundle about the medullary rays, but here in place of medullary rays we have the conjunctive tissues. So we'll write about the conjunctive tissues. So basically only the name is different. There is nothing different about the medullary and the conjunctive tissues. They are also the same extensions of the pith. So conjunctive tissues are also the extensions of the pith, and since pith is always parenchymatous in structure, so conjunctive tissue will also be parenchymatous. So it is also parenchymatous. So it is also parenchymatous. And if you see the medullary ray, it was also parenchymatous, and even this is parenchymatous. And lastly we can write that it is lying between the vascular bundles. So it is also lying between the vascular bundles. So conjunctive tissue and medullary ray, if you see, the medullary ray was also the extension of the pith. It was parenchymatous and it lied between the vascular bundles. So conjunctive tissue is also the same thing, only the name is different. And lastly we write about the central region of both the root and the stem, which is the pith. So about pith, so pith, we can write that it is the central region. So the central region, and yeah, we can even write the pith, it is formed of parenchymatous cells. So the pith, it is central region and it is made up of parenchyma. That's all about the pith, very similar to the pith of the stem, nothing different. And with this we complete the description of each and every structure of the monocot root, and only the figure is left. So about the TS I told you. Now we'll draw a portion of a TS which is enlarged. So if this is the TS, we'll draw, this is epiblemma, the root hairs are present, and then we can see two layers which are the endodermis and the pericycle, and finally the pith is there. So the vascular bundles are present. These are the xylem, these are the phloem, xylem, phloem. Okay, more than 6 in number. I drew this smaller, so only 4 I drew. So labeling, you can do root hairs, epiblemma, the cortex, the pith, the vascular bundles, the pericycle, the endodermis. So now we are making a portion of it enlarged. If we cut this somewhat like this, the phloem is also there. If we cut it like this, so what kind of structure it will be? So when we draw this enlarged, first of all we find that the root hairs and the epiblemma is present. So epiblemma, it is a single layer of cell. We know that a single layer of parenchymatous cells. So draw the parenchymatous cells, single layer, slightly elongated, and yeah, in between draw the root hair. Now don't draw the cells here. Do not draw the cells here because it is unicellular hair. I told you, in only the stem there are multicellular hairs. So draw 2 or more than 2 hairs here. So this is the epiblemma, and below the epiblemma we have the cortex region, which are formed of oval and rounded parenchymatous cells. So you can do it anyhow, oval or rounded. Okay, draw it as much as you can. Do not leave this intercellular spaces. There are very less intercellular spaces in the parenchymatous tissue. So this is the cortex, this is epiblemma, and this is the root hair, and these are the cortex. And now below the cortex lies the barrel shaped endodermis, which is the last layer of the cortex. So barrel shaped, make it somewhat like this. So we are taking this whole, okay, we are cutting it this way, not in this triangular shape. We are cutting it in this way. So when we cut it in this way, we find the whole circular structure here. Okay, so we will find the whole circular structure. This is the epiblemma, sorry, this is the endodermis. So draw the whole circular structure of the endodermis, and now below the endodermis lies the pericycle. Now pericycle, it is also made up of parenchymatous cells. So draw the structure here. These are also parenchymatous cells, slightly bigger than the endodermis. Draw the pericycle. So these are the Peri Cycle. This is the Endodermis and this is the Peri Cycle. Now between the Peri Cycle, inside the Peri Cycle lies the vascular bundles, but alternately. Now remember that xylem is exarch type. First of all, we'll make xylem. So xylem is exarch type. So which type of xylem will be in the central region? The metaxylem will be in the central region, because this differentiation exarch, endarch, centrarch and mesarch, it is on the basis of the location of protoxylem with respect to the metaxylem. Now exarch means that protoxylem lies towards the outside, the position of the protoxylem with respect to metaxylem. So exarch means the protoxylem towards outside, so metaxylem towards inside. So first of all, we'll draw the smaller protoxylem. They are the smaller protoxylem, and then make it bigger. This is the metaxylem. You can draw it more than 6, as much as you can. Okay, it depends upon your figure size. So in between we need to draw the phloem. Now we know that we can draw the phloem anyhow. You can just draw some of the structures like this. You can even draw some sieve tubes and companion cells, and then phloem parenchyma, and make it darker for phloem fibers. I told you in the last video as well. So draw the phloem in between all of them. So these are the metaxylem, these are the protoxylem, these are the phloem, and yeah, now the pith is left. Pith is parenchymatous, so draw parenchymatous cells all over. Just cover this all, parenchymatous cell. So this will be our pith, and this between spaces, the spaces between them, they are the conjunctive tissues. So this is just a rough figure, but when you see the clear figure, you will find somehow like this. So if you see the clear figure, you'll find it like this. The root hair is there, the epiblema is there, the cortex is there, the circular rounded cells. Then the barrel-shaped endodermis is present. You can see the barrel-shaped endodermis are there, and then the pericycle is present. And yeah, see, outside are the smaller protoxylem, and inside is the bigger metaxylem. You can see the phloem is drawn. If you zoom in, you can see that the phloem, it is drawn darker, and somewhere it is drawn just lighter, just to ensure that these are the phloem fibers, these are the sieve tubes, these are the companion cells, somewhat like this, just for differentiation. You can just draw it simple, a patch of cells as well. And then you can see the pith is there, parenchymatous, and in between there are conjunctive tissues. So this is all about the figure of the monocot root. And then when we go for the points of distinction, we'll write that after I will describe you about the Dicot root, because the points of distinction are clearly asked every time along with this question. So this is about the monocot root. Now I'll tell you about the Dicot root, and then we'll go for the points of distinction between both of them. So one more thing to remember, that the difference between the stem and the root. Most of the times you can get confused between the root and the stem. So once again I'll clear all the differences between the root and the stem, starting from the first. See, epiblema. In stem we had the epidermis, and the epidermis had multicellular hair, epiblema has unicellular hair. Okay, cortex is similar. Endodermis is very different from the endodermis of the stem. Though it is also the last layer of cortex and barrel sept, it is also thickened, but the reason of the thickening is different. The stem had the starch grains, but this root, it has the Kasparian strips, which is the lignin and suberin, deposition of lignin and suberin, the Kasparian strips, which do not let the water to enter, and in between some cells are there which lets the water to enter, known as passage cells. It lets the water to enter towards the xylem. And then pericycle, it is almost similar. Only one thing, that the pericycle of the stem, it was both sclerenchymatous and parenchymatous. Here it is only parenchymatous. Then we have the vascular bundles. Vascular bundles completely different. In the stem we had conjoint, we have radial hair. In stem the xylem was endarch, here the xylem is exarch. Phloem is almost similar. Conjunctive tissue similar to the medullary, only the name is different. Conjunctive tissue and medullary. Pith is everything similar to that of the stem. And with this we complete our monocot root. So basically the anatomical structure of the dicot root, it is very and almost similar to the anatomical structure of the monocot root. So I'll not tell you in detail about this. Just see, if you draw the structure of the TS of the root, dicot root, it will just, the thing different will be that this epiblema, it will be slightly thickened, This is epiblema. In monocot root we do only single layer of epiblema. This is the epiblema, and it is the pericycle, and then the endodermis, and then the pericycle. So vascular bundles, here the vascular bundles are arranged in ring shape. Suppose this is the xylem, then here is the phloem, again xylem, and here is the phloem, somewhat like this, extended type. Okay, in a ring shaped it is present. And so rest of the things are similar. Yeah, so we can even call it hypodermis, but it is, we do not write about the hypodermis, so let's call it the thickened layer epiblema. So I'm not trying to make you confused. We do not need to draw this figure as well. In examination we never need to draw the TS, we only need to draw the stem or roots portion which is enlarged. We never need to draw the TS, so do not get confused, do not take much time in this. It's just to understanding. The simple thing is that, as I taught you in monocot root, write about the epiblema, the same thing, everything same, nothing different, each and every word same. It means that it is also the outermost layer which is without cuticle, and it is thin walled, it consists of some unicellular hairs. And then about the cortex, again write the same thing, it is the layer lying below the epiblema, it is parenchymatous, and it is thin walled as well. And then again you can write about the endodermis, the very same thing as I told you in the monocot root. It means that it is the innermost layer of the cortex, and it consists of single layer of barrel shaped cells. And yeah, it has Kasparian strips and passage cells as well. The same thing you can write as we wrote in this in monocot root. And then we go for pericycle. The pericycle is completely same, we can again write the same thing, it is thin walled, it is parenchymatous in structure, and it lies below the endodermis and above the vascular region. And then we write about the vascular bundles. Even the description of the vascular bundle is same. It means that radial type, and the xylem and phloem, they are arranged alternatively. But now the difference comes, the first difference comes, it means it is arranged in ring. In monocot root we wrote that it is radial, xylem and phloem alternately but at different radii. It means radially it is arranged, it is arranged radially. Now what is the difference between radially and ring type? If you see the monocot root, a xylem was here, okay, and the phloem, it is lying here. And again if you see, the xylem will be lying here, and the phloem here, xylem here, phloem here. It means it is arranged at the radius, different radius, almost equal radius it is lying, so it is known as radially. But if we see the dicot, on the dicot root, you'll see if the xylem is present here, suppose this is the xylem, then the phloem is somewhat extended like this towards the another xylem, and then again the phloem is here extended, xylem, and again the phloem is extended. It means if you see that it is somewhat ring type in structure. So we do not need to define what is ring type and what is radially in the description, but I'm just making you understand. So basically we'll write it is radial arrangement, but it is in ring type. And yeah, the xylem and phloem, they are lying alternately. Now we'll write about the xylem. Yes, about the xylem, when we are writing, we will write the same thing. It is exarch, the protoxylem towards the periphery and the metaxylem towards the center. And then it is composed of tracheids, vessels, xylem, parenchyma, xylem fibers. And then about the phloem, now here comes the second difference. The phloem, it is also similar to the phloem of the monocot. It is made up of sieve tubes, companion cells, phloem, parenchyma, and etc. But here is the major difference, that above the phloem lies a band of sclerenchymatous tissues. So this is the thing to be remembered in the dicot root. It means when we make the phloem, suppose this is the phloem, then above the phloem, suppose this is our pericycle, okay, and this is our phloem, then above the phloem here lies the sclerenchymatous patches. It means first sclerenchymatous patch is there, and then the phloem is present. So above the phloem lies the sclerenchymatous patch, and this is about the vascular bundle. The only thing to remember about the dicot root is that the phloem has sclerenchymatous patches. So do not get confused between the root and the stems, it's very simple. And then about the conjunctive tissues and the pith, write the same thing as we wrote in the monocot root, nothing different. The conjunctive tissues, same thing, parenchyma, pith is also parenchyma, leave that, nothing different. And yeah, now with this we just need to draw the figure of the portion of the stem, portion of the root enlarged. So even here we cut it like this. So when we cut it, the structure will also be almost similar. First of all, we'll have the hairs, unicellular hairs, and this is our epiblemma. Here are our cortex. Okay, the cortex is there, and then we find our endodermis, the barrel shaped cells. Endodermis make a circular structure. Below that make the pericycle, it means parenchymatous pericycle. And then now make the xylem. First of all, xylem is exarch, so metaxylem towards the center. So metaxylem towards the center, and between them first of all make the sclerenchymatous patches. Now how to make sclerenchyma, I told you in the very first video, make double walled. So make sclerenchyma first in all the spaces, and then draw the phloem. These are our darkened phloem. Okay, we're expressing it like this. So phloem, and in between pith are present. So this is the basic structure. If you see the structure, it will be somehow like this. The very same, only here sclerenchymatous patch is there and phloem is present above it, and it is very similar to the structure of the monocot root as well. So with this we complete our both dicot and monocot root. Just the points of distinction, you can see it is almost similar, so no much points of distinction are present, but some of the points of distinction you can see that the sclerenchymatous patches are present. The first difference, points of distinction, if you write, so first of all number, points of distinction, and then monocot and dicot. Now this will not have more than 2-3 points because it is almost similar. So first of all, number of vascular bundles is the point. Number of vascular bundles, you can see the vascular bundles in the monocot. I told you in the monocot it is more than 6 plus, but in this, if you even see the figure, you can see 1 2 3 4 and 1 2 3 4. It means 4 are there, so it is not more than 6. It is usually less than 6. If we count xylem are 4 and then phloem are also 4, not usually more than 6. Usually in rare cases it can be, but in monocot it is more than 6 and it is less than 6 almost. And yeah, how can you forget about the arrangement? So arrangement of vascular bundles in this monocot radially, and in this dicot ring type. Now one last thing is the difference of the pith. I didn't tell you about this in the figure. If you see both the figures, this is my rough figure still. If you see the pith, it is larger in monocot, but if you even try to draw the pith here, it will be very small because all the ring type structure will cover almost all the portion, so only a small region of pith is present. So that small region of pith is present in the dicot root, so it is very small, and in monocot it is large. Even in some matured plants, the pith of the dicot, it is vanished. There is no pith in dicot in some of the plants. It means it may or may not be present, but in this monocot it is always present. And more than this, there are no points of distinction between the monocot and dicot root. So again, don't get confused between the root and the stem. I told you previously the differences between root and the stem, so you can rewind the video if you got confused. And with this we complete our both the anatomical structure of the root and the stem. So for now, goodbye, take care, wait till our next video.